Okay, good afternoon, everybody, and good morning for those of you on the West Coast. We're really excited to have another good group here for our third virtual research showcase series, which is again for those of you that are returning, and I see lots of familiar names, which is great. Part of a series, quite a long series that we've got planned for you to bring researchers and research project results that are being conducted across Canada with a variety of industry partners and academic organizations, all under the Cluster 3 funding project that's administered by the Dairy Farmers of Canada. Today, we're going to jump into best practices for silage production, essentially digging deep into what the research has to say in the context of this. For those of you that don't know me, my name is Stephen Roach. I'm a member of the ACER consulting team who's helping to coordinate the event series with a number of partners, most notably the Dairy Farmers of Canada. We are going to have more of a traditional approach today where we're going to have a great presentation by two of our researchers. We're going to have an opportunity for some open discussion as well as things go. So again, just reminding everyone, this is a normal or traditional Zoom room. In other words, you can take yourself off mute. You can put your camera on or off. We'll ask that when you're not speaking that you keep your camera or your audio rather on mute just to keep the audio line clear. We will be looking for comments and questions from you as we go today. So please, if you have any technical difficulties or any questions for the researchers, please use the chat feature. Within the chat, you can directly message my team members, Carly or Julia, and we want to be able to get your feedback so that when we have the question and answer period, we can dig right in. I believe we've covered most of the housekeeping except for one important item. If you are interested in listening to our presentation today or our talk in French, please navigate towards the bottom of your screen. There, just beside where you would share your screen, you're going to see the globe icon, which is interpretation. Click on that button and then choose the French channel, and also please choose the option to mute the original audio. That's going to allow you to get the best listening experience here in French. And if you're in English, of course, stick with us and we will take you on through. If you have any issues or other technical requirements, please just let us know. We'll do our best to troubleshoot those as we go. Otherwise, I think we're ready to roll. So again, just a big thank you to everyone for being here. Excited to be able to share this research with you. And I will hand things over to Amy for a quick comment from DFC.
Oops. Excuse me. Bad timing. So thank you, Steve, for the introduction. My name is Amy Desilets. I am Assistant Director Research at Dairy Farmers of Canada. I want to thank you all for registering for today's webinar. As mentioned, this is the third of a new webinar series launched by DFC just a couple of weeks ago as part of our ongoing commitment to research, communication, and KTT. This webinar series aims at showcasing all the exciting work happening in Canada. That is happening in dairy research across Canada. So this initiative is possible thanks to the financial support of DFC, but as well as our multiple partners. I also want to thank the research team today and all the other research team across the country for their collaboration under this initiatives. And finally, just thanking ACER Consulting for essentially organizing everything with this webinar. So without further ado, I'll turn the floor over back to you, Steve.
Thanks, Amy. So I'm going to bring in my other colleague, Julia Saracini. Julia is going to give us a little bit of an overview of our research team today, and then help walk us through the research as well. So Julia, please take it away.
Thanks, Steve. Hi, everyone. Thanks for being here. So I'm going to introduce our researchers today. So first we have Nancy. Nancy is an associate professor at the Department of Plant, Food, and Environmental Sciences at Dalhousie University. Her research spans topics including forage legume breeding, pasture renovation and management, low-cost soil and crop improvement, plant pollinator interactions, riparian zones, and plant tissue culture. She'll also be joined by Linda, who is a research scientist in plant pathology at St. John's Research and Development Center in Newfoundland. Her recent work has focused on topics including northern agriculture and season extension, diseases of forages and silage spoilage, and diseases of vaccine berries. So with that, I'll hand it over to our researchers to begin their presentations.
Thank you, Julia and Steve and Amy. This project, Linda and I are co-PIs on the project, but on the next slide, you can see that the research was done by many people. So Linda and I as co-PIs, but then important collaborators, Carol Lafreniere, who was immensely helpful in this project and conducted much of the research. Case Blazier and Kim Ominski from Manitoba, Shabtai Bittman in BC, and Emmanuel Uridwe here at Dalhousie University. And of course, graduate students, Katie McCollum, who has graduated from Dalhousie on this project, and Carla Platt, who is still in progress on her master's at University of Manitoba. Fred Waddy was a really important industry advisor on this project, and then there are many others, including research assistants and associates and technicians and provincial specialists, including Dave Dykstra, many people that were involved in this research, and some of them will be still going out and collecting silage samples tomorrow here in Nova Scotia. Catherine Rutherford and Ed McDonnell, they're just getting ready to sample again tomorrow. Next slide, please. Background, I'll say that successful milk production requires good forage crop management. I believe that's a dogma. I don't think I have to actually justify that. And that high quality silage from perennial forage crops can reduce costs of production, benefit cows, consumers, and the environment. And I have to disclose some of my biases. I live on a dairy farm in Nova Scotia, so I care deeply about cows and dairy industry. I've also been passionate about perennial forage crops for decades, since my very first semester as a student at Nova Scotia Agricultural College, and then later at McDonald campus. I have all of my research really has focused on perennial forage crops, whether it be in riparian zones or for feeding pollinators or for feeding ruminant livestock. Next slide, please. Some of the objectives of this research, so it was a 5-year project, and not all of the objectives will be covered in today's presentation. But for today's
Tout l'ensemble des recherches ne seront pas présentées aujourd'hui, mais nos objectifs principaux consistent à identifier les facteurs qui contribuent à un ensilage de grande qualité et à comparer les coûts annuels et les marges brutes dans divers systèmes d'ensilage. Ça, je vais en parler rapidement. Et avec Emmanuel Uridwe, avec Alvacan ici à Dalhousie, nous en avons parlé. Et le troisième objectif de cette recherche est d'évaluer l'ensilage afin de déceler la présence de microbes et de mycotoxines. Diapo suivante. Concernant la méthodologie, nous nous sommes centrés à récupérer des échantillons d'ensilage de luzerne sur l'ensemble du pays, en particulier en Colombie-Britannique, afin, je pense, de réduire le nombre de variables, tout simplement, nous concentrant tout d'abord.
First harvest silage from alfalfa and alfalfa grass mixtures. And we, so far, we're still collecting samples. The project is not over yet, so this is an interim report. 107 from bunker silos, 57 from conventional tower silos, and 79 from wrapped round bales. A few samples were collected from other types of silos, like drive-over piles and egg bags, but we were primarily concerned with 3 types of silos. Next slide. This slide just shows the in vitro dry matter digestibility at 48 hours, the range for bunkers, towers, and wrapped bales. And it's just sort of kind of raw data, but it's showing that the min and max were not very different for the 3 different types of silage systems. And the mean was, again, not very different. If anything, I would have expected wrapped bales to have a lower digestibility, but in fact it didn't. So in terms of digestibility, they were all quite similar. Next slide. Factor analysis was done in order to identify certain quality parameters that best described the different silage systems, I guess. And the factors which were identified that affected different parameters were legume content, not surprisingly, harvest date, again, not a surprise, the type of the silo, the use of a conditioner on the mower, and whether an inoculant was used or not. Next slide. With legume percent, this was done based on a calculation. What we found when doing correlation analysis between the calculated percent of legume and quality parameters where there were a number of significant correlations. Oops, could you go back, please? Thanks. It's a busy slide. So percent legume, decreased butyric acid, that's a good thing. Don't want a lot of butyric acid. It also decreased neutral detergent fiber digestibility at 30 hours. That's not surprising because neutral detergent fiber digestibility is higher in grasses than in legumes. So as legume percent increases, NDF digestibility is going to decrease because there's a lower percent of grass. ADF was decreased, good thing. Ash was increased, probably because alfalfa is high in minerals. Crude protein increased. It's a legume, so it's expected to have higher crude protein. And non-fiber carbohydrates went up, TDN went up, pH went up. That's because alfalfa has a high buffering capacity, so it takes more acid to drop the pH of alfalfa than just about anything else, even more so than other legumes. Ammonia went up. Alfalfa is kind of notorious for that. Lignin went up, a little bit surprising maybe, and digestibility, in vitro digestibility of dry matter went up. Next slide now, please. With harvest date, of course, it was also correlated with a number, not all, but a number of quality parameters. Ash went down as the date went up. Crude protein went down. So as the crop is maturing, the percent crude protein is reduced. Digestibility also goes down as you get later. So the harvest dates ranged from late in May to late in June. And first cut or first harvest, that is when the grasses are producing flowering stalks, flowering stems. And as you move through June, there's a higher percentage of stalk or stem and a lower percentage of grass. So that's why the digestibility decreases. Every dairy farmer knows that, I believe. The lactic acid went down, so there's less acid produced from more mature forage. Fiber went up, lignin went up, pH went up, and butyric acid went up. So that's not a new story. Harvesting late will reduce quality parameters of silage. Next slide. I was really interested in silo type significant effects. And I know there's a lot of information on this slide, so I put little stars on it, sort of highlight the main differences. So the stars are positive impacts on quality. And what I was surprised with was wrapped bales come out on top for quality compared to bunkers and towers. So that was a surprise to me. It was a surprise because I teach forages and cover crops class. It's one of my classes. And I've always told students that wrapped bale silage is less expensive than bunkers or towers where you have storage structures. And the quality might not be as good. So it's less expensive, but it's harder to make good quality wrapped bale silage. But in this case, we were sampling across the country, and when there were differences in quality, generally the wrapped bales had higher, for example, digestibility, which was a surprise to me. The pH is higher, but Is that more important than digestibility? Probably not. And there was also lower bound protein, which is good, although that matched the bunkers. So too much on there to talk about the whole slide, but let's jump to the next slide. Mower conditioners, when I teach forages, I tell my students that a mower conditioner will help to accelerate drying of stems, including alfalfa stems. So it speeds the drying. Speeding drying is a good thing. However, I was somewhat surprised to see that the use of a mower conditioner was not beneficial. So when there were differences in quality parameters, no conditioner was always better than using a conditioner. And that's an important difference. They're all, though, it's quite easy to explain because the different parameters are all looking at fiber and digestibility. They're all related. So what it's really saying is that you're losing leaves. And that's what, so with a mower conditioner, it's a compromise between speeding up the drying and keeping the leaves on the alfalfa. I thought maybe where it's silage and not hay, that you wouldn't lose as many leaves, but it seems like on average, we were losing alfalfa leaves when we used a conditioner. No conditioner has the other advantage of less horsepower required. You don't need an extra piece of equipment on your mower. You can potentially go faster, which has other benefits. So this was almost a pleasant surprise. Next slide, please. Inoculant is something that, well, I thought a lot of farmers would use a bacterial inoculant with silage, but in fact, in the samples that we obtained where we had survey information provided by producers saying whether they used inoculant or not, there were actually 80 that didn't use inoculant and 71 that did. And I guess going back, although we were able to test 243 samples, we did not have survey information on all of those samples. So some of these numbers are smaller than the 243, and that's because we are lacking information related to their management practices from some regions of Canada and some producers who did not provide it. So the use of inoculant was beneficial as expected. We had a lower pH, so more acid produced, had more lactic acid, which is what the inoculants usually do. Reduced butyric acid, that's important. So that would provide potentially better intake and a higher digestibility. Next slide, please. And I'm talking out of my area at this point with the economic viability analysis. This was conducted by, as I said earlier, Emmanuel Yeridwe and Hanbal Khan. And we based this on calculations for 150 kg butterfat daily quota, which was approximately the median for this survey data that was provided. And just the hard, I guess, conclusions were that the annual costs were, as expected, lowest with wrapped bales, highest with conventional tower, and intermediate for a bunker silo. And the gross margins were, gross margins is I have to be careful here because this is just the gross margin considering only costs associated with silage production. It doesn't consider all of the other various and important costs associated with producing milk on a dairy farm. So this is only taking out the cost related to silage production. So the highest gross margin was for wrapped bales and the lowest for tower silos. This is incredibly important information for farmers, maybe young farmers who are entering or farmers looking to expand. And this, though, has to be kept in mind that we're only looking at a small to medium sized dairy farm, 150 kgs, with larger farms, wrapped bales just don't make sense as the sole silage system anyway, but wrapped bales could be considered as extra overflow in some cases. We did not calculate the costs related to drive over piles, which would be another type of system. Next slide, please. And this is Linda.
Great, thank you. I'll take it from here to tell everyone a little bit about some of the work that we did to take a look at the fungi that were associated with silage samples. And so to do that, when you'd like to know what types of fungi are associated with just about anything, there's two very general ways that you can do that. You can take your sample and throw it on a Petri plate. And the good news when you do that is you'll end up with something that's living and growing on a Petri dish. And so that means it has to have been alive in the first place. You have something that's alive that you might be able to use in future studies. The downside is it's really time sensitive. It's also really labor intensive. We did some of this work within the project. You can imagine COVID threw a bit of a monkey wrench into some of our work related to this. And so I'll really be focusing on the DNA-based fungal detection methods that we used. And so we have a DNA sequence and a barcode over there. If you can, thank you, click my animation. So we mainly focused on this DNA-based detection method in order to understand what were the fungi that were associated with our silage samples. This is great because it tells us something about all of the fungi that were associated with the samples, but that means we were seeing information from things that might have actually already been dead at the time of detection if the DNA hadn't just had the opportunity to be broken down yet. And we're also really at the mercy of the quality of the data set that we're comparing to, because when we get DNA sequences, it's just a whole bunch of gobbledygook of letters. You need to compare that to something. And sometimes just through a quirk of evolution, you end up in a place where you might only be able to go so far in terms of the level of detection that you're able to do, or your ability to identify something to, the level of species. And we'll see that a little bit more as we go on. So click to the next slide, please. All right, so Nancy already told you about the samples that we collected. So we collected silage samples from across Canada. Those same samples were used for chemical and nutritional analyses, for DNA extractions, and also for the mycotoxin analyses that I'll tell you about in a few moments. And so we were only able to perform the DNA extractions and sequencing on the samples from the first sample collection year, so from the 2018-2019 samples. And once we had our DNA extractions done, we sent those for sequencing, and we analyzed the data in order to look for trends. So for example, to understand whether, we were observing certain fungal species associated, for example, with tower samples in Nova Scotia that we never saw in, bunker samples from British Columbia. So click to the next slide and we'll see what we saw. So I mentioned our methods were biased towards looking for fungi. And so I'm glad to say primarily fungi is what we saw. We obtained over 6 million DNA sequences that told us something about 4,000 different organism species. It gets a little tricky sometimes with this type of work. As I mentioned, it was primarily fungi that came out in our analyses. We also saw some DNA that snuck its way in that came from plants and from other things. So, the odd unfortunate grasshopper that might have happened to be in there. There was a little bit of insect DNA that made its way in. But of course, it was the fungi that we were most curious about because we were curious to know what was there and whether there was any possible association with mycotoxins. So next slide, please. And so this is what we found. I can accept, I guess, that most people are maybe not as passionate about fungal taxonomy as me. And so just to give a bit of an overview of what we're looking at here, we were curious to understand sort of what types of fungi was it that we saw. When you're looking at fungi, it's kind of similar if you're looking at animals, you have reptiles, you have mammals. So with fungi, the biggest groups that you have are ascomycetes and basidiomycetes. And so it's no big surprise that those two biggest groups of fungi were the two most highly represented in our data. So most of the fungi that we saw belonged to the ascomycetes group. The second most prevalent group belonged to the basidiomycetes. And within the ascomycetes and basidiomycetes, we were curious to understand kind of how it shook out in terms of what type of lifestyle those fungi lead. Do they tend to grow as yeasts, or do they tend to grow as filamentous fungi? And filamentous fungi grow as sort of like a hairy type growth form. And the reason why we were interested to understand that breakdown was because, generally speaking, filamentous fungi are going to be the group that can potentially include your mycotoxin producers. Yeasts, generally speaking, are not as likely to be associated with mycotoxins. They tend to be associated with risks of anaerobic, or sorry, excuse me, with aerobic spoilage. So that means perhaps a signal that oxygen was getting into these samples. And so generally speaking, filamentous fungi were what we saw among both the ascomycetes and the basidiomycetes. So next slide, please. I mentioned that we looked into various trends, and so we were curious to understand whether, again, for example, were there some fungi that we only found in association with, for example, silage samples that had a high pH versus a low pH, silage samples that came from a bunker as opposed to a bale from one part of the country versus another. We performed a wide range of comparisons in our data, and at the end of the day, generally speaking, no, we didn't see any major trends standing out to us. The one exception was that within Atlantic Canada, we did see some differences in which species were present, primarily in bunkers versus bales versus towers. And what it is that we saw was that some particular species were, we'll say, overrepresented in the tower samples as opposed to the bunker or bale samples. And so I'll get a little bit into what some of these species are and what type of things they might have been up to in those samples. So we'll move to the next slide. I mentioned that we saw over 4,000 different things living in there, and I won't sit here and take you on a tour of every single one of them, but just to give you some highlights of some of the fungi that we saw in association with our samples. The single most prevalent thing that we saw was a fungus that belongs to the genus Menascus. And that was one of the cases where, unfortunately, it's just a bit of a quirk of evolution that the level of DNA sequencing that we did, we weren't able to be certain which species of Menascus this actually was. Menascus ruber is a species that has been previously found to be associated with silage in other studies that were performed in other parts of the world. And Menascus ruber can also produce the mycotoxin citrinin. We can't say for sure whether that was the Menascus that we observed as the most prevalent thing in our study. It is a possibility. The second most prevalent thing that we observed was a fungus called Pichia anomala. Mycologists, who are the people who study fungi, love to give fungi all kinds of different names because as we learn more about them, we realize there's relationships we didn't appreciate. That's why there's a second name there for that organism. That was the second most commonly observed thing that we saw. And as I mentioned, it's a yeast, and it has also been found in association with silage in other colder parts of the world. So not a huge surprise and kind of reassuring to us to see that, the same types of fungi that we see here in Canada are also things that people struggle with in other parts of the world. The third most common thing that we observed was a fungus that belonged to the group Penicillium. I don't think I need to tell people that things that belong to the group Penicillium are among some of the fungi that produce some really interesting chemicals, including, of course, penicillin, which can be a good thing under the right circumstances. But some species of Penicillium produce chemicals that we wouldn't want to find associated with silage, including mycotoxins like patulin and mycophenolic acid. Penicillium is also something that has been previously found to be associated with silage in other parts of the world. So next slide, please. Thanks. So these are some of the fungi that we found that were more highly prevalent in the tower samples within Atlantic Canada, as opposed to in the other storage types. So what were they? So the first was Deberomyces hennessii. That's a yeast that is associated with extreme environments. And when we're talking about extreme environments, unfortunately, we're not talking about like super cool fungi like with skateboards. We're talking about extreme things like, for example, very low oxygen type conditions, which of course is what we would expect to find in silage ideally. We found the fungus Cladosporium was overrepresented in those samples. And once again, that was a case where we weren't able to be certain which species of Cladosporium we were dealing with. But we know that Cladosporium is a filamentous fungus that can be associated with silage and plants in general. Leucosporidium is another yeast that can be affiliated with extreme environments. And finally, Candida was one of the other fungi that we found in those tower samples. And some Candida are associated with plants. Some can be associated with, human and animal, infections. There's no indication that that was the type of Candida we were observing here. Certainly, it's likely to be a plant-associated species. So next slide, thanks. All right, so now I'd like to tell you just a little bit about the mycotoxin analysis that we performed. And so what we did, if you click to my next animation here, thank you. For all of the samples that were collected in this study, we submitted them for analysis at a commercial lab, and they screened for a panel of 16 different mycotoxins. So if we move to the next slide, This is what we saw. So as I mentioned, we analyzed over 200 samples for 16 different mycotoxins, and I'm very pleased to say that generally speaking, we didn't see a lot of mycotoxins. And so that's good news for everyone whose samples were included in the study. So what did we see? We saw the mycotoxin zearalenone present in 1 sample. We saw mycophenolic acid present in 16 samples. So that was the mycotoxin that we saw the most of across everything. We saw deoxynivalenol present in 1 sample, and we saw sterigmatocin present in 4 samples. And so at the end of the day, out of those 200 plus samples, just over 20 of them had any mycotoxin at all that was present above the level of detection. So that doesn't mean that there were no other mycotoxins possibly present. We weren't able to look for everything. We only looked for 16 specific mycotoxins in this study. And as well, at the end of the day, analytical equipment can only tell us whatever it's able to detect. And so it's possible that other mycotoxins were present or that these were present in a greater number of samples, but we weren't able to detect them because of their very low concentration. But we were pleased to see that there was a relatively low incidence of mycotoxins, and where we did observe them, for those mycotoxins that have a maximum safe limit determined for feed by the CFIA or by the USDA, the levels that we observed were always below those levels of concern that have been established. So that's good news that mycotoxins were not highly prevalent in our samples. So if we move to the next slide, these are conclusions that pertain to the work that Nancy's presented.
Thank you, Linda. And just going from what Linda was saying, some of the samples that we will be collecting this fall will be tested for a wider range of mycotoxins at a lab in London, Ontario, Mark Samara's lab, I believe. So that's great news. And that came about due to some communications from those researchers. Conclusions that high quality silage, luckily, can be made using different silage systems. We knew that, but that I think producers shouldn't stress too much about different systems, that all systems can make good quality silage, as long as the producer is informed about what's required for good quality silage. And that wrap bale silage can match or exceed the quality of higher cost systems, including bunkers or tower silos. That a mower conditioner use, at least in the study, did not improve silage quality. So perhaps that should be something that when new equipment is purchased, that a conditioner not be included. And Dan Undersander in Wisconsin has been pushing this for a number of years, that it's more important to spread the freshly cut forage out in a thin layer on the ground rather than to use a conditioner. So it really just backs up some of what Dan Undersander's been saying for a number of years. That inoculant use probably isn't a waste of money. It's a fairly inexpensive management technique that can provide better fermentation. And in terms of the economic analysis, and I have to also qualify this. So the economic analysis was based on 150 kgs of quota, but it was also based on a 60% forage dry matter in the ration, and the forage was only perennial grass legume mix, so there was no corn included in the calculations. So when corn is included in the calculations, then that completely changes the cost, because if you're growing corn, then you may need to use a silo harvester. So most of the differences in the cost were related to the reduced equipment costs. But if you're making corn silage, then you would need that equipment anyway. So that would completely change the cost analysis. So this was based on no corn silage. And we know a number of farmers don't grow corn silage, but a number do. So this was assuming no on-farm equipment for corn silage as well. And yeah, so based on those assumptions, the wrap bale silage was less expensive and higher gross margins when you're considering just the cost of producing silage. Next slide for Linda.
Thanks. So when we looked at what fungi were associated with silage samples from, again, different storage types and from all across the country, we found a very wide diversity of fungi, including some that are sort of the usual suspects when you're looking at silage samples, and some others that hadn't been previously reported from silage. We didn't find any major trends in terms of fungi that were associated only with one particular storage type, or for example, when you had or had not used an inoculant, with the exception of the fact that within Atlantic Canada, we did see some differences with respect to the representation of some species, particularly in tower samples as opposed to in bunker or bales. When we looked for mycotoxins, we screened for a panel of 16 different mycotoxins. And although we did see some mycotoxin associated fungi within the fungal sequencing data that we looked at, generally speaking, the incidence of mycotoxin detection was quite low. And where it was detected, it was below the limit of detection, or excuse me, below the level of concern where a level of concern has been established. And so with that, we'd like to move to the next slide. We'd like to, of course, thank you very much to the Dairy Cluster for funding and support of our research project. And of course, an enormous thank you to Canadian dairy farmers who participated in this project, who filled out surveys, who allowed producers and others to come on farm to collect samples. And of course, a big thank you to all of the other members of the research team, especially the grad students whose very hard work was behind a lot of the work that was presented today.
That's wonderful. Thanks, Nancy, Linda. Really interesting results. Really exciting to see so much work being done in this area, and to hear more work is coming as well. So really appreciate you walking us through that. We do have some questions, so I'll read some of those out now. But I'll also just note anyone listening, if you have additional questions, just pop them in the chat for us, and we'll be happy to address or bring those forward as well. Nancy, a question for you right off the hop. You adjusted a little bit in your conclusion, so maybe you can build off of it or see where you'd like to go with it. And initially, it was around how economics actually factor into the discussion around maybe decision making for those younger farmers. When you sort of stacked up the results there in your conclusion, you said rat bales really seem to yield good or maybe even excellent results, at least based on some of the work you've done here, and seem to, if I recall that initial slide on the economics, have the highest gross margins. I know that there's an asterisk around that in terms of that calculation, but is that a fair takeaway? And I guess if I'm a young farmer listening out there, is this for sure where I should be going, or is there an important caveat to that in terms of maybe how people could make decisions around where they go or what silage approach they take?
Well, yeah, all kinds of, so all the systems that we evaluated were good. And normally a farm doesn't start from scratch. So normally you're going into an existing farm, taking over an existing farm, or joining family members in a farm. So I think using what you've got makes sense, what's there. But if you are looking at increasing your forage production, increasing your getting more quota, and you are at capacity for the silos that you have, I think that perhaps round bale silage makes sense. If you were in an unusual situation where you were going to start a new farm, then I think round bale silage makes sense, as long as it's only a small to medium sized business. If it's a large dairy farm, then bunkers, I think, makes sense. There's economies of scale, but there's also, in order to manage that much forage quickly, then you're going to be needing to go faster, bigger equipment. So yes, I fully support round bale silage. And personally, frankly, my son is a dairy farmer, farming with his father, my husband, and he expanded field crop production when he joined the farm in his mid-20s, and he frankly decided to go with wrapped round bales. We've tried a number of different silage systems at home, so we have some experience with all kinds, and it seems to be working out well. And when he was doing that, I frankly didn't have a good answer for either my students or for him. Is this the way to go, or should we build a bunker? along with the towers. And now I feel confident in telling farmers that, young farmers especially, that wrapped bales are a very good silage production system. They're less expensive. I also like the fact that perhaps they're less fermented, so they're retaining more forage, more quality, more energy. And in some cases, wrapped down bales have been shown to provide superior effects, growth on beef animals. So I'm not the first person to say that wrapped round bale silage can be good. It was a long answer. Sorry for that.
That's great. Thanks, Nancy. It helps to peel back the onion on the thinking around the genesis for this project too, which is pretty cool. A question that maybe applies to both of you, and so maybe just to keep things fresh, Belinda, we can start with you. Both of you talked a lot about the culmination or the synthesis of the data that you've collected. I wonder, are there geographic differences that are important, or regional differences that are important or interesting perhaps to speak to? Does geography actually come into play based on results in terms of quality, what we can expect, or in terms of mycotoxins or microbial results? Linda, any thoughts there?
Yeah, so that was certainly something that we were looking to try to understand over the course of this study. And at least when we looked at the data for the fungi and for the mycotoxins, that was actually not something that jumped out at us. And to me, that was a big surprise. I did expect to see some regional differences just because, for example, you're going to have different fungi living in different parts of the country, and, where you have different summer temperatures, or, producers are better able in some parts of the country than others to dry the forage in the field before it gets ensiled, just because of, what Mother Nature is going to be throwing at them while they're trying to make that happen. We certainly did expect to see some differences in the populations or in the incidence of mycotoxins, but that actually wasn't something that we observed. And so it might just be because of the heterogeneity of the project as a whole, and just, farm to farm differences sort of evened out some of those differences, which made it difficult to, point to any one, for example, species of fungus that was present in one place and not in another. And with the mycotoxin detections, because we were lucky to not observe a whole lot of mycotoxins, ultimately we didn't have the statistical power to be able to say, this is found here and not found over there. And so no big trends with mycotoxins, to be sure.
And the same with the quality parameters. We did not find many differences. We really didn't find any significant differences between regions. That was an initial objective of the study, to see if there were differences in different regions, but we didn't find any. The alfalfa was the legume chosen for this project because it is a legume that can be grown across Canada, although the British Columbia samples were mainly just grass, but all other provinces provided alfalfa grass mixes. It's not necessarily my favorite legume, but it was chosen because not all places can grow the same legumes, but alfalfa is adaptable across the country. So yeah, really no differences in regions, which was surprising, but I think it was a positive as well. So the results can be applied widely rather than just within regions.
Yeah, agreed. That's really cool. Linda, just going back to you for a moment. Obviously, decisions need to be made in all research projects about sort of the bounds of how far can we go with resources and what's possible and all of those details. You mentioned some of the limitations in terms of how deep you could go, or how broad a spectrum of tests we might be able to apply. Would you do anything differently next time? And I guess if the answer is no, because you've captured it really nice, you made good choices there, where might you be headed next in terms of thinking, helping to deepen your understanding of this aspect of the research?
I think it's always a little dangerous to ask a researcher what they would do with more time and more funding. Of course, there's always more that we would like to do. So I mentioned that, for example, the DNA sequencing data pertained only to the first year's worth of samples. It would be great to have access to that, data for the second year of samples. There's a bit of a shipping issue with the DNA involved, and so unfortunately it wasn't able to be recovered. We weren't able to to get that same level of detail for the second year of data, but that would have been excellent information to have. It would have also been nice, I mentioned that we looked primarily at fungi, and that was because we were curious to understand whether we might be able to detect mycotoxins and therefore trace it back to, ultimately what was responsible for those mycotoxins. But bacteria also, of course, play a very important role in the microbial community in silage. And in a perfect world, it would have been amazing to have the opportunity to take a look at the bacterial community alongside the fungal community. And I also mentioned, and it's great, as Nancy mentioned, there's going to be the opportunity to do some further studies with relation to the mycotoxins, because where we were somewhat limited by, what was available to us, through the commercial labs that we utilized for the analyses, it would have been great to be able to have a wider scan for mycotoxins among our samples. And it was just a bit of a quirk of the timing of the project. And it's excellent to know that there'll be the opportunity to take a bit of a deeper dive in the future.
Yeah, that's great. And Linda, I'll stick with you for a moment because we've got another question. And then Nancy, I'll ask a similar question just in terms of forward thinking a bit. But we had a question specifically on, reviewing some of those conclusions around the levels of mycotoxins. And surprisingly, at least it was for me, seeing that they're quite lower than the harmful level. Would you think that mycotoxin binders, so the purchase of them and the use of them, would be justified? Any thoughts there? I know you didn't maybe explore that specifically, but just pragmatically thinking.
Yeah, that's an interesting question. And as you said, that's not something that, was, included as, a scientific objective in this study. With the very low levels that were detected, and with the information that we have in front of us, I think that would be a difficult decision to make. I suppose I would be curious to, see the results of further studies, because again, at the end of the day, we were only looking for a relatively narrow scope of mycotoxins. And so there could be something in those samples that would be something of more concern, where a mycotoxin binder would indeed be a very worthwhile investment. But with the information that we have, I would say it didn't jump out at me as a very significant concern, given, the concentrations and the frequency with which, we did observe mycotoxins.
Yeah, that's fair. So Nancy, jumping to you, I guess I'll ask both questions. So things you might do different next time if you were to, just reflect back on how the project's unfolded, and if nothing else, thinking forward, how do we help to deepen some of our understanding here? What more might you do? Where are you headed?
One thing that I would do differently is I would provide a less comprehensive survey to the producers, so that we could, it really was, there was really too much information we were initially looking for. And as a result, some producers did not provide information on their management practices. So we had their silage, but we didn't have information on what their management practices were. We knew the type of silo, but we didn't know whether they used inoculants or not, or conditioners or not, or their harvest dates. So I would not overwhelm them with a comprehensive survey. That's one thing. Going forward, my interest is really in different legumes. So this one was just, we only took silage if it was alfalfa as the sole legume in a mix. But I think that it would be nice to compare alfalfa with other legumes that have less soluble protein, to look at what happens to that in a silo. So we know that some legumes hold on to their protein, it's not lost as quickly, or solubilized, or lost as ammonia. So I'd like to compare alfalfa with other legumes that are probably underutilized. And I'd also like to look at different grasses. So this was just grass. We didn't look specifically at any type. We didn't put any restrictions on what grass species were included. So I think there really needs to be more work looking at different species compositions for silage, different grasses, different legumes, mixtures. I think, but it's difficult research to do, but I think it's important to be done.
That's great. One final one just from me, and Linda, or Nancy, sorry, just going back to that comment about where this came from, this research came from. as you share this with your partner, with your son, with some of the other folks that, perhaps you had some initial conversations about, hey, how do I craft this project? Any surprises from them? Any sort of take home messages that you're just hearing as you talk with them about some of these results, or have they already been shared here, you think?
They're probably not. They're probably not joining here. I would say that, yeah, my son, he knew what he was doing. He did actually take my forages and cover crops course. That's not why he knew what he was doing. No, he made a really good decision. And I guess I certainly wasn't doing this just for my own family. I was doing it so that I would have more informed information to provide for my students. So I teach every year, I teach students who will be going to beef farms or dairy farms or sheep farms. They're going back to their family farms or to join other farms. And I was never really confident in saying that wrap bale silage is as good as other types of silages. I knew that it was less expensive to make, at least in a shorter term, but I really didn't know, and I didn't know the economic story behind it. So yeah, I felt like I needed to be more informed so that I could give more knowledge, informed decision making for future dairy farmers. And I'm passionate that dairy farmers need as much support and as information that they can apply as possible. So I hope I answered your question.
That's great. It's very cool to see how the questions or the research sort of came from grassroots, and I think more and more we benefit from the inclusion of producers in some of those initial and very pragmatic questions, and then start to build around that. So it's just really exciting to hear. You don't often get to hear where these things come from sometimes. So I think that's great, and it really cool to see how the results have come to fold here, especially through pandemic and everything else is not without its challenges. So Nancy, Linda, really want to thank you again for taking the time to share with us today. We'll be very excited to share further results as they come out, work with your team to develop some other outputs, and share this recording as well for others that may not have been able to join here today. So thank you again. For everyone that's on our call, just hang on for one more minute. I'm going to bring back my colleague, Julia. She's just going to briefly comment on where we're headed next in terms of our virtual research showcase, and then and some final comments before we wrap up.
Thanks, Steve, and thanks again, Nancy and Linda, for your excellent presentation. Thanks, everyone, for being here today. Just to touch on the upcoming webinar that we have on November 16th, so just in a couple weeks, we'll be featuring Dr. Herman Barkema and his research team as well. So same time, 1 to 2 p.m. Eastern, again, similar style of a Zoom meeting. And so Dr. Barkema will be featured in the 4th session of our showcase to learn about antimicrobial use, antimicrobial reduction, infectious disease surveillance, as well as latest research on outdoor access. So we really hope that you're able to join us. This is also the information in French as well. So we will be promoting this through emails similar to this research showcase as well, starting next week. If you're interested, please feel free to subscribe or directly message one of the team members here, and we can have you registered for this event as well. So thanks everyone again for being here today. It's great to see some familiar faces. And again, thank you to Nancy and Linda and your research teams. It was great to have you, and we're looking forward to seeing you at the next one.
Thanks everyone. Have a great afternoon.